Good morning to all. Welcome to the Raymond Group of Institutions NEET and K-CET online classes. Today's topic is cell cycle and cell division. So in the first question is proteins and RNA are synthesized in dash phase of cell cycle. So we know that cell cycle and cell division, the sequence and the duplication of the genome and the formation of the daughter cell from the parental cell is called as the cell cycle and cell division. In that cell cycle and cell division, here proteins and RNA are synthesized in dash phase of cell cycle. When we can see as a cell cycle, it's having two phases. One it is M phase and another one is interphase. Here the interphase is called as a resting stage or resting phase. The resting phase or interphase again subdivided into three phases. Those are G1, S, G2. So here G1, S and G2 phases and M phase again classified into two karyokinesis and cytokinesis. After the completion of the cytokinesis, G0 phase will be formed. See here, but the question was asked proteins and RNA synthesized in the phase of. So this is the complete cell cycle and cell division to the formation of two daughter cells from the single parental cell. You can follow into the two phases of M phase and the interphase. Here the interphase is also called the resting phase. It contains three phases as G1, S and G2 phases. So here means of the G1 phase. G1 phase is also called as gap 1 phase. In the gap 1 phase, different types of the proteins and different types of the cell organelles or duplication takes place in a G1 phase. Means different types of the proteins and cell organelles and also the RNA molecules or duplication takes place or synthesizing takes place in a G1 phase, because here after the G1 phase, S phase we are called as synthesis phase. In that S phase, duplication of the DNA, what amount of the DNA it was present in a cell, that amount of the DNA in S phase will be going to replicate or it will going to multiply, or they are also called as double. And after the S phase cell, it will be going to enter into the G2 phase. Means in the G1 phase, what are the proteins are not synthesized and what are the proteins and enzymes and cell organelles are required to the M phase, those all are again synthesized into the G2 phase. So here the question is asking proteins and RNA are synthesized, means initially which stage proteins and RNA is synthesized. So options are given as G1, G2, S and none of these. So we know that G1 phase, G2 phase and the S phase. G1 gap 1 phase, G2 is gap 2 phase and S phase, synthesis phase in the S phase only. Duplication of the DNA it takes place. But the question asking as a synthesis of RNA and proteins in S phase. Initially RNA and proteins are synthesized in a G1 phase only. So answer is G1 phase. And the next question, when cell has stalled DNA replication for which checkpoint should be predominantly activated? So at the time of cell cycle and cell division of the eukaryotic cells only they can follow the cell cycle and cell division. So the time of the completion of cell cycle and cell division, two checkpoints are we can see. So those are the two checkpoints. One of the checkpoint it was present in G1 and S phase. In between the G1 and S phase, one checkpoint will be known present here, checkpoint 1, and second checkpoint it was present in between the G2 and M. Here the checkpoint 2 it will be known present. So what are the checkpoints? Checkpoints will be going to maintain the CDKS, cyclin dependent kinases proteins. Cyclins are a special type of proteins they require into the synthesis or they can maintain the kinases enzymes. So by the involvement of the cyclin dependent kinase enzymes, they can maintain the cell cycle and cell division in a sequence order. So first checkpoint or CDKS checkpoint 1 it was present in between the G1 phase and S phase. This is also called as G1 checkpoint. And the second checkpoint it was present in between the G2 phase and the M phase. So now here question was asking when a cell has stalled DNA replication for Here DNA replication is take place in a S phase. If you are going to stop the DNA replication in a S phase, if you are going to stop the DNA replication in a S phase, which checkpoint should be predominantly activated? Means here first checkpoint it was started in a G1 phase and it will be going to complete or inactive at the S phase. Second checkpoint was started in a G2 phase and it will go to complete and inactive at the M phase. So now here S phase DNA replication is take place. In the S phase replication 4 it will be going stalled, means we are also called as stopped. So which phase otherwise which checkpoint should be predominantly activated? Means before the stopping of these replication 4, which checkpoint will be going to activated? Means here checkpoint 1 it was present into the G1 phase and it is inactivated into the S phase. Means before the inactivation only, when we are going to start the DNA replication it was stopped means automatically second checkpoint it should be start. The second checkpoint should be as a G2. It should be a presence in between the G2 phase and the M phase. So when we are going to stop the DNA replication in a S phase, it was started a predominant checkpoint. It is in between the G2 and the M phase only. That's we call as a G2 checkpoint. So options are given as here. Option 1 G1 and S. Option 2 G2 and M. Option C M, and option D both G2 and M. So here the second checkpoint it was present in between the G2 and M only. When we are going to start the DNA replication at the S phase, so from the onwards second checkpoint it was started. That second checkpoint it was present in between the G2 and M. Otherwise we are also called as a G2 checkpoint only. So that's why answer is A. And next question: the DNA content of individual cell and the number of cells in each phase of a cell cycle can be determined by using flow cytometry. So cytometry is an instrument we are going to use into the measuring of the DNA in each phase of a cell. So which are the following combination of phases of cell cycle and its corresponding DNA content can be considered as a normal? So here question is asking, if you are going to measure the DNA concentration in every cell of different phases by the using of cytometry, which phase of the cell cycle will be going to consider in a normal? So option 1 diploid cells found in the G2 or G1 phase. So here again this portion belongs into the cell cycle and cell division only. Then you can draw as for your easy way of understanding. Cell cycle and cell division will be followed by the two steps as interphase and the M phase. Interphase again subdivided into G1 S and G2 phase. M phase is contained as a karyokinesis and the cytokinesis after the G0 phase. So here cell it was diploid cells found in the G0 or G1 phase. Diploid cells are found in the G0 or G1 phases. See here now, always cell cycle and cell division it will started from the G1 phase only. The cells, the diploid cells always found in the G1 phase or the G0 phase, because from here onwards cell cycle and cell division it started. And option 2, cells with the twice the normal DNA content in the early M phase. See now, cells with the normal DNA content in the early M phase. This is the M phase. So before the M phase, G2 phase is there. Before the G2 phase, S phase is there. So what's the major difference between the G1 phase and G2 phase and S phase? So in the G1 phase and the G2 phase, major difference it is. Here the G1 phase, different types of the RNAs and proteins are there synthesized. And in the G2 phase, what are the remaining enzymes and proteins are required for the M phase? Those are synthesized. In between the G1 and G2 phase, S phase is there. So what happens in the S phase? S phase, it's called as synthesis phase. Here the DNA replication will be going to takes place. What's the amount of the DNA, the copy number of the DNA, it will going to increase or duplicated from the G1 phase into S phase. Chromosome number is constant, but the amount of the DNA only doubles in the S phase. See now, cells with the twice the normal DNA content in the early M phase. Yes, cells with the twice normal DNA content in early M phase. When we are going comparing to the G1 phase, in the M phase or early M phase, amount of the DNA, we are not asking about the chromosome number. They are mentioning as a amount of the DNA, that is DNA content will be going to double in a S phase. So that's why same amount of the DNA was seen in the G2 phase, and same amount of DNA we are seeing in the M phase also. So that's why here when we are comparing the G1 phase and the early M phase, amount of the DNA will be going to double in a M phase. And option 3, cells have the half amount of DNA in the G1 phase as compared to the G2 phase. Yes, already we have discussed here in the G1 phase and the S phase. S phase DNA replication take place, amount of DNA will become multiply. If you are taken as here, in the G1 phase, amount of the DNA it was having 2C. Here the C represents as a copy number. In the S phase, amount of the DNA will be going to become as a 4C. And the G2 phase, amount of the DNA will be going to present as a 4C. Now you see, cells have the half amount of DNA in the G1 phase as comparing to the G2 phase. Here the G2 phase 4C amount of the DNA is there, means 4 copy number of the DNA is there. But when we see into the G1 phase, it's a 2C only, half of the amount of DNA only we are seeing into the G1 phase than in the G2 phase. And option 4, cells with twice the normal DNA content in the G2 phase. Yes, cells with the twice and the normal amount of the DNA will be going to present in the G2 phase compared to the G1 phase. So what are the given options? Based upon the cell cycle and cell division, as regarding G1 phase, S phase and the G2 phases of amount of DNA. So in the alien options are there, we have selected the correct options. 1 and 2 are correct, 1 and 3 are correct, 3 and 4 are correct, and 1, 2, 3, 4 are correct. So based upon these options, amount of the DNA from the G1 phase, S phase and the G2 phase is mentioned. So these are the 1, 2, 3, 4. 4 options are there correct about the cell cycle and cell division and the amount of DNA. Answer it is B. Next question. Which of the following statement is not correct with regard to G0 phase? So in the G0 phase we are also called as a quiescent phase. In some of the animal cells, example as a heart cells, the cells are then not continue the cell cycle and cell division. They will enter after the M phase into G0 phase or quiescent phase. Basically after the M phase into the cells are they have entered into the G1 phase. But these are the heart cells they will enter from the G1, the M phase into the G1 phase, and they will not continue the cell cycle and cell division. They will going to return back into the G0 phases. So these type of the cells are they have replaced by the one another, and at the time of damaging of the cells, most of these eukaryotic cells and some of animal cells are they can enter into the M phase into G0 phase, G1 phase into G0 phase. So G0 phase is a resting phase, but the cell it was metabolically active, and after the some period of the time, again this G0 phase into cells will be going to enter into the G1 phase, and they can enter into the S phase, and later they can enter into the G2 phase, and they can continue. But at some specific period of the time only, some of the cells, that is example heart cells, they will unable to continue the cell cycle and cell division. And some of the cells they can return, they can enter from the G1 phase into the G0 phase. The G0 phase is the quiescent phase or G0 quiescent phase. In the G0 phase, cells are metabolically active. So in the they given as the cells becomes metabolically inactive. They are written as a metabolically inactive. Which of the following statement is not correct with regard to the G0 phase? They are asking, means out of the 4 statements for the G0 phase, which statement is incorrect statement? So now here the cell becomes metabolically inactive and the cells functions as a reserve cells. Here cells are there enter from the M phase into the G1 phase. That's a normal cell cycle and cell division. But some of the heart cells they can enter from the G1 phase into the G0 phase, means that is called as a reserve cells. And option C they given as the cell can join cell cycle at any time. After the some period of the time, at any time, again these are the cells that can enter from the G0 phase into the G1 phase, G1 phase to S to G2 phase. So they can continue the cell cycle and cell division at any time, and most of the cells leave G1 phase midway and enters into the G1 phase, G0 phase. So most of the cells are leave G1 phase, means from the G1 phase into cells are they can enter back into the resting state, that only we call as a G0 phase. So out of the given statement, which of the following statements are not correct regarding of the G0 phase? So B and C and D are correct statements for the G0 phase, and A statement only here they given as a metabolically inactive. Cell cannot perform the cell cycle and cell division continuously, but these cells are metabolically active. So answer it is A. Next question, find correct statements for cell cycle. Find the correct statement for the cell cycle. See here, cell cycle means a sequence arrangement of the cells growth and the duplication of the DNA and cell organelles, and then form into two daughter cells from the single parental cell. That's called as cell cycle. So in these are the cell cycle sequence arrangement. Otherwise, they have to find out the incorrect statement of cell cycle. So option A is given as cytoplasmic growth is continuous process. Yes, cytoplasmic growth is a continuous process. So at the time of cell cycle and the cell division, to the formation of two daughter cells from the parental cell here, what's the amount of the cytoplasm is there? That amount of the cytoplasm it should be continuously increased. So that is called as a continuous growth of the cytoplasm. And option B, DNA synthesis continues to occur all the time. Here in the cell cycle and cell division, DNA synthesis or replication take place at one time only, that one phase only, that phase it is a S phase. So option B is given as DNA synthesis continues to occur all the time. So it will not take place in the all time, it can take place in the particular stage of the cell cycle and cell division only, that is interphase of S phase only. It's not take place in the all stages. In the G1 phase, G2 phase, DNA replication will not take place in the S phase only. But the option B is given as DNA synthesis continues to occur all the time. And option C, nucleus is larger during the cell division. Yes, at the time of the formation of cell cycle and cell division, once the nucleus is there, the nucleus size should be large. At the time only, karyokinesis will take place easily and they will form into a daughter cells. And specific stage observes replication of DNA. So that specific stage observes the replication of DNA is a S phase. So here find the incorrect statement for cell cycle. So cytoplasm increasing take place continuously, and the nucleus is large at the time of cell division. Specific stage only DNA replication take place. And the incorrect statement is B, DNA synthesis continues to occur at all time. It's not at all time, at one specific stage only, that is the S phase. So answer is B. Next question, select the correctly matched pair. Option A, an astral meiosis. Example they given as a higher plants. Yes, meiosis. So cell cycle and cell division, it is two types of the cell divisions are take place, that is mitosis and meiosis. Mitosis is a equational division, meiosis is a reduction division. So they are asking about the meiosis, an astral meiosis in higher plants. So higher plants in the mitosis and meiosis, in the both times karyokinesis take place. So in the plants, a specialized cell cycle and cell division take place at the time astral rays and centrioles and astral rays are not found. So that's why we call as an astral meiosis. Example as higher plants. And option B they given as a microtubules. They given as example are matched photophosphorylation. Here the photophosphorylation means synthesis of ATP molecules. Phosphorylation means ATP molecules, photo means light. So through the photosynthesis ATP is synthesized. ATP synthesis, that is called as a photophosphorylation. But here this is not a correct match for microtubules. Microtubules are made up of by the specialized protein. The protein we call as a tubulin. And spindle fibers. Option C, they are given as a spindle fibers. Spindle fibers means at the time of the cell cycle and cell division, chromosomes are there attached by the presence of a small thread-like structure. That thread-like structure is only we call as a spindle fiber. That spindle fibers are made up of carbohydrates. So they are given as a carbohydrates. It's not made up of by the carbohydrates. Those are made up of by the microtubules. So, and option D, none of this. Out of the given options, correct match is anastral meiosis is an example higher plants, and astral meiosis example into the animal plant, animals, and also the animals that form centrioles at the time of cell cycle and cell division. So correct match is A. And next question. In zygote, homologous chromosomes undergo pairing to form bivalent. These two chromosomes are attached laterally due to. Here, concept of these meiosis. So meiosis occur by two stages: meiosis 1, meiosis 2. Meiosis 1 again having two types, that is karyokinesis and cytokinesis. The karyokinesis again occur by the four stages. Those are prophase 1, metaphase 1, anaphase 1, and telophase 1. These are you know. So in the meiosis 1 of prophase 1 again having as 5 substages. That those are leptotin, zygotin, packetin, diplotin, and diakinesis. Now you see here the question is in zygote, homologous chromosomes undergo pairing to form the bivalents. So bivalents are formation take place, or bivalents are we are seeing into the meiosis 1 of the prophase 1 of the zygote stage. So in the zygote stage what happens? Here in the zygote stage cells are having homologous chromosomes. So which are the chromosomes are they are similar to each other, those are we call as a homologous chromosomes. So these are the homologous chromosomes. So in the zygote what happens? These are the homologous chromosomes are they are paired. So pairing of the homologous chromosome. This is the one chromosome and this is the other one of chromosomes. So these two chromosomes are there paired. Pairing of the homologous chromosomes is called as a bivalence. They will form as a bivalent. The two chromosomes are attached laterally due to. Here the two chromosomes are there attached, and they can presence in the specialized structure as nodule-like structure. That nodule-like structure it is option A, U protein, helicase protein, ribonucleoprotein, and endonuclease proteins. So here two homologous chromosomes are the non-sister chromatids are paired together by the presence of the specialized structure, that is ribonuclease protein. So here U protein they given, and helicase protein, ribonucleoprotein, and the endonuclease protein. So endonucleases are the specialized structures that include the DNA inside, and ribonucleoproteins, these are the specialized proteins are they hold to the two non-sister chromatids of homologous chromosomes. So this is only a button-like structures, and helicase enzymes that include the DNA in between the two strands of the DNA. Hydrogen bonds will be going to present, the hydrogen bonds will be going to break down by the helicase enzyme. And U protein we are going to tell as it is used into the animals. These are the U proteins will be going to be European as a racil. So now here the specialized proteins are they will be going to used to bind the two non-sister chromatids of homologous chromosomes, that is at a bivalent stage. That protein is called as ribonuclear protein. And here pairing of the two homologous chromosomes that called as a synapsis and forming a bivalence, and also they can form into a tetrons. Here the four chromatids are there involved 1, 2, 3 and 4. So answer is ribonucleoprotein. And next, what will be the amount of DNA in a haploid cell which had 10 picograms of DNA in its nucleus after G1 phase? See now here we have to identify the amount of DNA. Amount of DNA in a haploid cell which had 10 picograms of DNA in its nucleus after the G1 phase. Again, we have to draw the cell cycle and cell division. M phase, interphase, G1 phase, S and the G2 phase. So now here hint it is amount of the DNA in a haploid cell is a 10 picograms of the DNA in the nucleus after the G1 phases. So we have to identify the amount of the DNA. Here after the G1 phase, amount of the DNA is 10 picograms in nucleus. Means we have to identify the amount of DNA in haploid cell. So a haploid cell means cell that have entered into the G1 phase only. So amount of the DNA will going to multiply in S phase. If the amount of the DNA is having in the S phase is 10 picograms, means in the G2 phase also 10 picograms, in the M phase also 10 picograms, but in the G1 phase amount of the DNA it will be going to present into half of the S phase. So 5 picograms of the DNA was present. So answer is 5 picograms of the DNA in the G1 phase. Next question. Arrange the following events of meiosis in the correct sequence. They are given as a crossing over, synapses, terminalization of chiasmata and the disappearance of nucleolus. So here meiosis they are given. Meiosis is a reductional division. In that meiosis 1, that 2 in prophase 1, leptotin, zygotin, patatin, diplotin, and diakinesis. So these are the 5 stages of expressing a meiosis 1 of prophase 1. It's a very complex stage of meiosis 1. See now, in the leptotin stage, chromosomes are undergoing as condensed, then appears clearly under the light microscope. In the zygotin, so in the zygotin what happens? Pairing of homologous chromosomes. So these are the homologous chromosomes. Homo means similar structures. In the zygotin it will be. So these are the zygotin. These are the homologous chromosomes will be going to pair. The pairing of the homologous chromosomes we call as synapses. This is called as synapses. And here these are the 2 homologous chromosomes are they paired together by the present involvement of ribosome. So we are seeing in a patatin stage. So now in the zygotin what happens? They can form into a pairing of homologous chromosomes that's called as synapses, and they can form as a bivalence. So these are the 2 chromosomes are called as bivalence, or they are also called as a tetrods. Tetrods formation in a zygotin stage. And next stage is patatin stage. In the patatin stage, after the pairing of 2 homologous chromosomes of the non-sister chromatids, in between these 2 by the involvement of ribonuclease enzyme or recombinase enzyme, so in between these 2 exchange of the genetic material will take place. So the exchange of the genetic material of the non-sister chromatids of homologous chromosomes in the presence of ribonuclear protein, this is called as a crossing over. Crossing over takes place. After the crossing over, that is occur in a patatin stage, later diplotin stage. In the diplotin stage, they can form into the repulsion of homologous chromosomes of the non-sister chromatids, and these are the entire chromosome will become separated only at the end of chromatids. They can present that connecting of the 2 chromatids. They can form as a X-shaped structure. That X-shaped structure only we call as a chiasmata formation. We are seeing diplotin. Later the diakinesis stage, 2 chromosomes will become separated. In these are the 5 stages. They have to identify the sequence order in meiosis. Means they given as crossing over, synapses, terminalization of chiasmata, and disappearance of nucleolus. So disappearance of the nucleolus is the final character of the prophase 1. So out of the 5 stages, first stage is the leptotin stage. After the leptotin stage, zygotin stage. In the zygotin stage, synapses take place. Means, so first it should be synapses, pairing of the homologous chromosomes. After the pairing of the homologous chromosomes, after the J-coding stage, next stage it is particling stage. In the particling stage, crossing over is take place. So after the synapses, second crossing over. After the crossing over, in the next stage of the particling, diploiding stage. In the diploiding stage, chiasmata formation take place. Means these are the ribonuclear proteins. They can move into the ends of the chromatids. They can form into chiasmata. This is the third. And last one is disappearance of the nucleolus. Means first synapses, crossing over, terminalization of the chiasmata, and disappearance of the nucleolus. So this is the first, second, third, and fourth. 2, 1, 3, 4. So now 2, 1. So answer is a 3. 2, 1, 2, 1, 3, 4. Here 2, 1, 3, 4. Answer is a D. Next question. Read the given statement and select the correct option. So statement A. Every chromosome has 2 chromatids. Every chromosome has a 2 chromatids. Every chromosome contains as a 2 arm-like structures. So see now, these are the 2 arm-like structures are we call as a chromatids. Every chromosome has a 2 chromatids, and DNA replication during the interphase. Yes, DNA replication takes place in the interphase of the S phase. S phase of interphase. So every chromosome contains a 2 arm-like structures we call as chromatids, and DNA replication during the interphase. So both of the statement A and statement B based on the option A. Both statement A and B are the true. B is the correct explanation for A. Here A represents as a chromosome having 2 chromatids. B represents as a about the interphase as a replication. So now both of the statements A and B is correct only, but in between the A and B does not have any relation. So B is not a correct explanation of A. Option A is incorrect and option B is check it is. Both A and B are the correct statement, but B is not a correct explanation for A. So here B says as a that is replication of the DNA that is take place in the interphase. Interphase having the 3 phases as a G1, S, and the G2 phases only. So in the S phase DNA replication takes place. So statement B also correct, but for the B and A does not have any relation. So B is not a correct explanation for A. So answer is B.